Right, so everyone, we have now finished with viruses. There's a few different things I would like you to do to finish up with, I suppose, corrections, and that'll be posted on the Google Classroom. But for now, we are continuing with bacteria. What we're looking at today really is the different types of groups that we classify bacteria into. We're looking at bacterial cells and their structure. And hopefully, we will see if we get there, but we will see if we can understand a little bit about the reproduction of bacteria. You probably know a lot about bacteria already. Kind of things we know about bacteria is that they are unicellular, and that means that they are single-celled organisms. So unlike us, we are multicellular. But bacteria, every single bacteria out there has only one single cell, and that makes them unique. Bacteria are also, I suppose, what we call prokaryotes, and we will look at this word in a bit more detail later on, but know that prokaryotic means that you don't have a normal nucleus. And that means that your DNA just floats about the cell. They don't have normal organelles because normal organelles have what we call a membrane surrounding the organelles. And bacterial organelles don't have that. And the other final thing to note as well that you should know at this stage is that bacteria are a very broad group of organisms. They can be either autotrophic or heterotrophic, and that means that some of them will produce their own energy. That means autotrophic, and heterotrophic meaning that some bacteria don't produce their own energy. They have to feed on other organisms. So bacteria are just an enormously abundant group of organisms. They are probably the most abundant organisms on our planet. They are broadly categorized by scientists, though, and you may have heard of some of these words before. So I suppose the words that I would like you to remember would be spherical, rod-shaped, and spiral-shaped. So physically, we look at bacteria and we describe their structure under what we see underneath the microscope. I see spherical. It's just like a sphere. And if you look at it, it looks spherical. We see rod-shaped. So it has this sort of horizontal, I suppose, rectangular sort of shape. So we call it rod-shaped. And spiral-shaped physically looks like a spiral. So of each of these different groups, then you should be able to remember an example of, we'll say, a spherical, a rod-shaped, or spiral-shaped bacteria. So the example of a spherical one, and you'll all have heard of this, or rather, you'd certainly have suffered its ailments. Streptococcus is a type of bacteria that causes sore throats. Streptococcus, and that is also a spherical type of bacteria. Bacillus typhoid, in other words known as TB, was a very harmful disease that affected people's lungs. And then again, that was a rod-shaped bacteria. And spiral-shaped bacteria, it would have included things like something called Campylobacter, which is an organism that causes food poisoning. So what I would suggest to you is just learn off we'll say the shape of the bacteria and an example of the bacteria. So spherical, Streptococcus. Rod-shaped, again, I gave you the example of Bacillus typhoid, or you could also pick E. coli if you prefer E. coli, and spiral-shaped Campylobacter. The next thing then is actually drawing a bacterial cell. Now, I would like you to look at the notes that I'll be posting and use the notes that I'm posting a more simplified, I suppose, accurate version of this diagram. But just in studying this bacterial cell overall, and we're looking at a rod-shaped bacteria, just like, I suppose, an animal cell, the bacteria does have some similar components. Like, for example, this line here, and you could call this number 4, represents a cell membrane. And on the inside of the cell membrane, you have cytoplasm. Cytoplasm here is kind of represented with the kind of creamy white color. And remember, that's just a liquid that chemical reactions take place in. We have these dots that represent the ribosomes. And we also have, I suppose, a cell wall here that's represented here. And that'd be something I suppose you'd be more, familiar with in finding one with, we'll say, a plant cell. So there are some, I suppose, familiar organelles, but there's also some that you wouldn't have seen before. Like, for example, on the outside of the cell wall, you've got something called a capsule, and the capsule kind of includes all of this part on the outside. And it's like a slime layer. The capsule is a special layer that's there for protection of the bacteria. Another thing then is the flagella. The flagella is like basically the tails that flow out of some bacteria, not all. Very similar, in fact, to a sperm cell, the tail you'd find in a sperm cell. And the flagella, of course, has a role in movement. The other part then, and these two parts you're probably looking at and saying, well, where's the nucleus? There is no nucleus, remember, because a bacteria is considered to be prokaryotic. And that means that a bacteria has no true nucleus. The nucleus is not membrane bound. There's no nuclear membrane. And that means that the DNA, or the single chromosome that makes up a bacteria, is able to float around and nothing stops it from just flowing anywhere at once. So this is just what you could call a loop of DNA, or you could call it a single chromosome. This is poorly represented in this diagram, but I'm going to pop over to notes in a minute, and you will see that this kind of loop here is what you call a plasmid. And a plasmid is probably one of the most important parts of a bacteria for defense, because bacteria need to be able to produce defenses against what we call antibiotics. In fact, they need to be able to produce their own antibiotics anyway, because bacteria produce antibiotics to kill other competitors. Antibiotics are a natural chemical produced by bacteria that kill bacteria. And all of this happens here as a result of this part here, which is called the plasmid, which is basically a loop of DNA that's responsible for the production of antibiotics, as I said. These parts and functions will be, I suppose, something that you could almost hazard a guess at. Like, it's pretty obvious, the flagella there, just like in a sperm cell, the flagella must have a role in we'll say, movement. The nuclear material, we're not going to call that a nuclear material, we're going to call that DNA. And DNA has a role in, of course, genetic inheritance, okay, in reproduction. You could talk about the ribosomes, of course. The ribosomes are there to produce proteins. There's nothing new there. The cell wall is for protection. Now, we're going to call this a cell membrane instead. We don't need to call it the plasma membrane. But the cell membrane, just like any other cell, it's controlling what enters or leaves the cell. The capsule is there for protection, so it's a slime layer involved in protection. And as I said there, the plasmid is involved in antibiotic resistance. So there's some bacteria that contain all of these cell organelles, but other bacteria don't contain all of them. So not all organisms, or not all bacteria contain flagella, for example, and not all bacteria contain capsules, for example. But again, I suppose these are the parts and functions highlighted. Right, asexual reproduction. For bacteria, they undergo, I suppose, a form of reproduction we call binary fission. Binary fission is basically where a bacteria clones itself. You start with what we call a parent bacteria. The parent bacteria will reproduce its DNA, so it replicates the DNA, it elongates, and it then becomes bigger and splits apart. We call the two cells that are produced as a result from that one cell the daughter cells. Now, I don't know will I get this going, but maybe we'll see. Pop down there. No. Right, this may or may not work, and it's going to turn off the sound. We don't need the sound on that. Not available. Right. Um, well, look, if we just describe through it and explain what happens. So you start off with a normal bacterial cell. Okay, you don't need to worry about all the different labels here. Just a few labels would be fine. Start off with one loop of DNA. The DNA, as I said, replicates itself. So now we've got two. The cell will elongate, in other words, it becomes longer, and the DNA will move to opposite sides of the cell. So this is one loop of DNA, and this is the replicated loop of DNA, we'll say. A cleavage furrow develops. A cleavage furrow is just like the burrow that develops in the middle of the cell. And eventually, these cells will split apart, and the cell wall will reform. And now we've got two daughter cells, which are themselves capable of reproduction. So bacteria do this type of reproduction, binary fission, very, very often. Every 20 minutes or so, many species of bacteria can reproduce. But the one thing about them is that they are complete clones of each other. Their DNA is absolutely identical. And that, I suppose, poses its own risk that their DNA is so similar. Now, the one thing though is because their reproduction is so quick, bacteria can actually mutate also very quickly. So you can imagine if every 20 minutes a bacteria reproduces, the population of bacteria becomes very big very quickly. And the more that these bacteria are exposed to antibiotics, the greater their resistance. But we're going to leave that for another day because I want to spend a little bit more time on antibiotics. So what I'm going to show you now is the work that you'll be doing for me for tomorrow. So we have got out of this now these notes. I'm just going to ask you to write up the definitions at the start of this chapter. Now, there is an awful lot of definitions, so many of which I haven't talked about today, but we will be coming on to. So get as far as facultative bacteria and also spend time learning them. That'll be your work for today.